I'm very much thrilled to have all of you on this session. Thank you all the speakers and attendees for joining this online event. I'm Mahesh Kumar Verma, working at Veeda Lifesciences Private Limited, a company focused on finding the new therapies for skin autoimmune diseases, including vitiligo. I'm your host and I'll be moderating today's session. I'm so excited to have all of you for our webinar session on vitiligo, treatment and beyond. And really looking forward for a very interactive session. I'm thankful to the Veeda Lifesciences and in particular to Dr. Parul Ganju for enabling this session. My sincere thanks to Ms. Vinodita Pathak and Ms. Nikita Ravi from Veeda for all the background work. This webinar is hosted by Veeda Lifesciences with the Association of Veeda Clinical Research, who tremendously helped us to make it streamlined and also to reach to the bigger audience. As you know, today is World Vitiligo Day, which is observed every year on June 25th. This is an initiative with the purpose to build global awareness about vitiligo. This is a disease which can have a significant negative social and psychological impact on patients. Vitiligo affects 1 to 2% of the world population, and it does not discriminate based on gender, ethnicity, religion, caste, or food habits. Vitiligo patches can appear any part of the body. Generally, it is considered that this disease is autoimmune in nature with some genetic predisposition, which means that body's own immune cells attack and destroy the melanin producing cell. The World Vitiligo Day started on the year 2011, and this year is its 12th occasion. World Vitiligo Day is also called the Vitiligo Awareness Day or Purple Fun Day, as purple is the color chosen as vitiligo awareness color. 25th June is chosen as a memorial to Michael Jackson, who suffered from vitiligo, and he passed on these days. In today's session, vitiligo treatment and beyond, keynote address will be delivered by Dr. K. Vijayaraghavan from NCBS, who was the principal scientific advisor of the government of India. Among the speakers, we have professionals who are expert in human clinical trials. We have expert clinicians who treat vitiligo patients on day-to-day basis. We have research scientists who work on the basic research or basic biology about the vitiligo. We have social activists who aware the people about this disease and allow for the inclusiveness to happen. And also we have the entrepreneur with us who help to find the new therapies for the vitiligo. So to start this session, first of all, let me invite Dr. Parul Ganju from Veeda Lifesciences for opening remarks of the session. Dr. Parul Ganju earned her PhD in vitiligo biology at prestigious National Institute of Immunology, Delhi. That's when she got passion to do something worthwhile for vitiligo patient, and that the idea led to the formation of Veeda Lifesciences. She plays both the role of scientist and entrepreneur and is the co-founder and the CEO of the company. All to Parul.
Thank you, Mahesh. It is said that confidence is the most beautiful thing that one can possess. This World Vitiligo Day, we urge everybody to embrace their differences and to stay confident in their own skin. We are all here today to make people understand what vitiligo is, to dispel any myths that are associated with the disease, and also to assure the patients that they are not alone in their struggles. With the support of a lot of clinicians, researchers, and now social activists, we are at a very exciting stage where a solution to this problem is really not a distant dream anymore. So what exactly is vitiligo? Vitiligo is a disease where white patches appear on the skin. The reason why this happens is because the color producing cells of the skin, which are called melanocytes, they get killed by your body's own immune system, leaving behind a white patch. This makes vitiligo an autoimmune disease. And unlike common perception, it's not a contagious disease. It doesn't spread by touching. Now, what really bothers the patients is that vitiligo changes their outward appearance. And added to this is the fact that it's an unpredictable disease. So patients have to live a life of constant fear. A fear of not knowing when or where new patches are going to appear. As Mahesh said, I got a chance to interact with a lot of vitiligo patients during my PhD research. And that's when I understood the problems that they have to oversee in the society. One story that has actually stayed with me was that of a girl who wasn't able to attend her own sister's marriage because of the misconceptions associated with the disease. Several other patients remained unmarried. They underwent severe psychological stress. We even had patients who had suicidal tendencies. So clearly, these patients have a very painful relationship with confidence. Now, as researchers, we had these poignant stories on one hand and the possibilities that our science had to offer on the other. That's when Dr. Natarajan and I decided to use our science to develop new and comprehensive treatment strategies for vitiligo patients, which would help us remove the fear and the stigma associated with the disease. This has been the guiding star for our startup company, which we set up in May 2016 and called Ahamune Biosciences. Ahamune is an amalgamation of the Sanskrit word aham, meaning mine, and immune from immunity. And together, this means my immunity. This name highlights our efforts towards trying to identify the solution to the autoimmunity in skin. In a way, our efforts are also related to the word confidence. The confidence that as scientists, we will be able to find a treatment for this no-cure disease, and the confidence that we would be able to change the perception of India from the generics capital of the world to the next drug discovery destination of the world. Now, over the last 6 years since our inception, and it's all thanks to the hard work and the perseverance of our entire team, and the support that we have received from our board members, advisors, and investors, that we have been able to achieve a breakthrough in vitiligo therapeutics. Our team has identified a new drug candidate for vitiligo treatment, which is both safe and efficacious. We call this drug candidate AB1001. And all our studies establish that AB1001 has the potential to not only stop the spread of the disease, but also to bring back color to these white patches, treating vitiligo. We are also very proud of the fact that the entire research and development for this molecule has been done completely in India. And we are now super excited to transition into a clinical phase company. And what better day to announce this transition than the World Vitiligo Day, which marks a new beginning in the life of vitiligo patients who are looking for something new in this area. Today, we take our first step towards our promise of providing a brighter future to the vitiligo patients with our phase 1 first-in-human clinical studies with AB1001. We are excited to partner with a wonderful team at Veeda Clinical Research for this study, about which you'll be hearing a little more about it from Dr. Martha. Just to give you a small introduction about Dr. Martha, I mean, he doesn't need any introduction, but he over the last 3 decades, he has been passionately involved in first-in-man clinical studies and has been helping companies like ours execute such studies. He's currently the director and member of the board at Veeda Clinical Research, and we are also fortunate to have him as our principal investigator for our phase 1 study. Over to you, Dr. Martha.
So, shall I share your slide, Dr. Mark?
No, I think I can.
Yeah, you can.
Can you see it now?
Yeah, we can see it. Please make it full screen.
Yeah, thank you very much, Dr. Parul, and warm welcome to Dr. Natarajan, Dr. Vijay Raghavan, Dr. Mahesh, and other distinguished speakers. It's a pleasure to be on this panel to discuss about the vitiligo and a very important condition of vitiligo. And I have an opportunity to share my pleasure of helping Ahamune in developing a very important molecule for vitiligo. So I'll just give you some idea initially with what Veeda is, because Veeda is partnering with Ahamune to take the molecule forward. It is like, they have Dr. Parul's group has done the discovery work and we are doing the development work. We are partnering in the development process. So just to give a very brief about Veeda CR, it's a clinical CRO conducting clinical trials from phase 1 to phase 4 clinical trials, and also the bioequivalence studies with corporate office located in Ahmedabad. We are one of the very major CRO conducting phase 1 studies, including first-in-man studies, a single ascending dose study, multiple ascending dose study, drug-drug interaction studies, food effect studies, etc. And we have 2 units of phase 1. One is of 18 beds, another one is of 12 beds, and both are approved by the Drugs Controller General of India. And we have a specifically trained staff available at both the sites for conducting phase 1 studies. We also collect certain PK samples at the time when we are doing the study. And we have something like more than 40 LC-MS machines. And we analyze more than 100,000 samples in a month. And we also are experts in doing some PD and biomarkers analysis through our another lab, which is a joint venture between Veeda and Sumru from Canada for large molecule. And we have a special relationship with our pathology lab or a safety lab, which is NABL and CAP accredited. We always work with the ethics committee for the safety of the subjects, and they keep a very close eyes on us. And Veeda have been inspected by the all the regulatory authorities of the developed countries, and also inspected by major pharma and biotech companies. And with this, I would like to mention that we are very excited to work with Ahamune in developing the molecule. This molecule we are going to try with a phase 1 open label dose escalation study, which is the first-in-man study for essentially to evaluate the safety and tolerability and aided by pharmacokinetics of the molecule. And as you know, phase 1 is essentially for the safety and tolerability, and we take all the possible care for the safety of the subjects. In this study, the plan is to have 4 cohorts of 3 subjects each, and we will move in that direction, and each cohort will receive a different dose as such. And of course, the study conduct wise, we will have the DCGI approval and ethics committee approval, and followed by there will be screening of the subjects as per the SOPs of Veeda. And just out of those screened subjects, there will be subjects who fit into inclusion exclusion criteria. They will be selected, and ultimately the treatment will start in them. But they all, as you know, they are all healthy subjects, because the phase 1 first in man is usually in a healthy subjects. But of course, we will also have the dermatologist who will check them before starting the treatment and also after completion of the treatment. And the investigational drug will be dispensed and applied to the subject's body in a defined area. And of course, the photographs will also be taken of the forearms, pre-dose and also post-dose, to evaluate if there is any concern so far as the local irritability or sensitivity is concerned. We will also carry out the clinical examination and the defined laboratory investigations. And after completion of each cohort, we have a DSMB meeting, so that the DSMB discusses if there is any safety concerns in the healthy subjects. And then once they approve to move to the next higher dose, we will go to the next higher dose as such after the DSMB meeting takes place. So as you can see, we take a lot of care in safety of the subjects. And as you can see, all these activities are involved in which we need to have a lot of coordination with not only with Ahamune, with dermatologists, with the doctors, with the cardiologists, with the intensivists. We always have an intensivist also present at the time of conduct of the study. So safety is of the utmost importance, barring apart the PK parameters and the skin condition of the subject. So it's a very proud moment for Veeda that we are associated and privileged to have an associated with Ammune and their dedicated scientists in their endeavor who are constantly thriving for quality, safety, and caring of the patients as such. So thank you very much. And once again, thank you, Dr. Parul and the Ammune group to give me an opportunity to give a little brief about how we would be conducting the study as such. Thank you again.
Thank you, Dr. Marthak. It's wonderful to hear about the phase 1 clinical trial. This AB1001 for the phase 1 is going, it's a topical treatments, topical treatment. So, Dr. Marthak, I'll just take 1-2 questions which has come to us about this trial. So, though we have a small question answer session at the end of the study, but there are some particular questions. So, just let me ask it. You rightly mentioned that phase 1 trial is mainly about that safety and the tolerability. So, and many of the vitiligo patients are very much concerned about that, the safety of the drugs, what they say, because many of the drugs has a lot of side effects. And this particular treatment being topical in nature and overall the nature of this molecule, where you place this new therapy, the AB1001, compared to any other in terms of the safety for the patient?
Depending upon the preclinical data that we have seen, I feel the product will be very safe, and I do not expect any kind of the adverse events or the serious adverse events with the molecule. But as we move forward, we will be able to judge, and then only we can mention about it. But on prima facie, based on the preclinical data, I do not envisage the serious adverse events. And this being a local application, the chances of the serious adverse events are very minimal. And so I don't expect anything.
Oh, that's very wonderful to hear. Very nice for the patient also who are here in our attendees list. There is one more question, like when we do the clinical trial and we say once the trial is successful, it will come to the patient, this drug will be available for the patients. So many people, they ask me, what does it mean by a successful trial? Right now it is the phase 1, slowly we'll move to phase 2 and phase 3. So from a patient perspective, what would be a successful trial mean?
For the patient's perspective, now every patient has the different kind of expectations. Somebody is worried more on the cosmetic aspect. Somebody is more worried about the disease aspect. So everybody's perspective is different. But so far as science is concerned, which is ultimately beneficial to the patients, is that the success is when we say it is safe for the patients, and it is efficacious. So once it is safe molecule, though it may take a little longer time to heal, but it is the best way of treating the patient. So safety is of utmost importance.
Yes. Thank you, sir. And people also are very excited and they want to know, see if everything goes fine, or the trial goes fine, by when they can expect this drug is available in the market.
Oh, it is a very difficult question to answer.
Let's hope that everything goes in.
But by and large, any product to come in the market, it takes at least 5 to 6 years minimum. And this type of condition, which is usually of a chronic in nature, it takes a little longer time to complete the study. So, but there are various ways we can discuss with the regulatory authorities, depending upon what we see in phase 1 study, depending upon what we see in the phase 2 study, also depending upon the early phase 3 clinical trial. If you're saying very good response, we can always go to drugs control authorities, tell them to give just like what is happening in Corona, is the emergency use authorization, but continue with the study. So one can design the way of moving forward as we see the results.
Okay.
So, but by and large, at least 5 to 6 years it takes before it comes in the market.
Okay, great. So there is hope for the patient in due course of the time. Thank you very much, Dr. Martha, for joining. And thank you very much for this update and exciting talk. Thank you very much.
Thank you so much.
Now I'll invite Dr. Natarajan, Dr. Krishnamurthy Natarajan. He is also a co-founder of the Immune Biosciences. Him and Dr. Parul started this company, which has now made the drugs AB1001, which is in the clinical trial. So I'll invite him to chair the keynote session. Please, Dr. Natarajan, over to you.
Thank you, Mahesh. Thank you very much, and welcome. Good afternoon to everybody. As others said, of course, one of the main purpose of today's webinar is actually to create awareness, really for a humane, sensitive, and sort of inclusive society for the millions of people who are actually suffering from vitiligo. I think Parul articulated some of those issues, but I think that is the prime purpose. And of course, in the process where Immune is pitching in to contribute our part, but that's a long, story away. But we have a good hope moving forward. Indeed, it's a great pleasure and a privilege for me to welcome and introduce Professor Vijay Raghavan. I think it is a wonderful, it is a terrific occasion for all the participants here to hear him, his wonderful achievements that is actually being flashed on the screen. I specifically, there are many things that Professor Vijay Raghavan is known for, other than the fact that he started off as a geneticist. Many of us who are geneticists have fond likeness to geneticists anyway, right? So, and then he actually did some fundamental work. He has been doing fundamental work in the area of neural circuits and muscle development using the fruit fly Drosophila melanogaster. His career started at TIFR and went to Caltech and came back to TIFR, and then eventually was one of the first people to have started the National Center for Biological Sciences, Bangalore. Many accolades. Of course, there are some of them indeed very, very special. For example, to have named gecko lizard after him by a discoverer of this is indeed a very, very special thing, and not many people have that honor for that. Of course, he has been known for as an institution builder and as a thought leader in the scientific community in the country, and truly he chartered the Department of Biotechnology from 2013 to 18. Following which he was the principal scientific advisor to the government of India. And this is a very important moment, period, I should say, where the country and the world saw the pandemic. His efforts as a PSA has been phenomenal, where among many things, what he came up was the National Research Foundation, which eventually will, of course, be fruitful at some point of time very soon, I suppose. And one of the quotes that I thought worth putting it here, among the many things that he said, is that with regard to the pandemic, he said, this is a time for us to repurpose all the research we do in the country, whether we wield a pencil or a radio telescope, to this focused task of managing or controlling or finding tackling this COVID-19 pandemic actually. He contributed enormously during that period, of course, and he continues to inspire many of young generations in the country, actually. And with that few words, I really am grateful on behalf of our Immune for him to have very readily accepted our invitation to address the audience today. Thank you very much.
Thank you very much, Dr. Natarajan. It's a great honor to be here today to hear about your company's enterprise and zeal. And it was very good to hear Dr. Kiran Marthak, Dr. Mahesh Verma earlier, and Dr. Parul Ganju. And I'm sure that we will have more discussions later on going forward. You are really experts in the area. And as I said earlier, I have nothing to offer by way of advice, except offer appreciation for the daring and courage you've shown. What I'll try to point out to the audience, however, is the extraordinary complexity and the risks in drug discovery and how courageous it is for you having taken it up. Now, our understanding, so I will divide my very brief talk into three parts. One is to first address the issue about how our understanding of biology has changed enormously. The second is, how does that impact on the interventions we make in various kinds of aspects of human biology, including lifestyle diseases, inherited disorders, and so on and so forth. And third is, where do we go from here in these kinds of areas of drug discovery and applications to treat diseases. All of this, of course, has suddenly taken center stage because of the pandemic, and that has brought biology to center focus in a manner which otherwise it wasn't there at all. And in some ways, that is the kind of both opportunities as well as challenges we face. Now, coming to our understanding of biology, it's rather dramatic to see how much things have changed over the last few decades. And that is because of a combination of genetics, biochemistry, as well as physiology. Now, this has come about in the following way, because biologists principally, as Sidney Brenner said, ask three questions. They want to know where it came from, that's evolution. They want to know how it is made, that's developmental biology. And they want to know how it functions, that's physiology. Medical people want to know how to fix it when it's broke, right? So that's really a kind of engineering aspect of it. And in medicine, you're not too bothered about understanding or knowing necessarily about the basics of biology. As long as what you do fixes it, you're fine. You must keep in mind, though, that biology, unlike in engineering, where there are very rigorous principles in engineering about what you can do and what you cannot do, and they stand the test of time. Medicine is much more of a hit and trial area where new understanding can have dramatic change in what you hit and what you try. For example, you can have an important cancer drug, which is very effective in 10% of the people who you treat, and 90% they're not. And therefore, you would normally have said that this drug is a very poorly effective drug. It treats only 10% of the people with a particular kind of cancer. However, if you have the understanding of mechanism of various cancers and which of those specific kinds of subsets of cancers a drug affects, then you find that it actually has an impact of 100% in or close to 100% in those kinds of cancers where it works. And you don't treat the other 90% unnecessarily. So an understanding of the biology is very important. But let us take these three components about evolution, developmental biology, and physiology each together. Our understanding of evolutionary biology continues to grow enormously, but it is not integrated into the space of the medical profession or in terms of the pharma sufficiently. The reason for that is the following. So Darwin and Wallace developed the theory of evolution by natural selection, and they fundamentally showed that all life on Earth is related through a shared chemistry, whether it's the earliest of single cellular organisms to the largest of animals we have now. And this shared chemistry is connected through the thread of DNA. In other words, what we've later discovered about evolution is a mechanistic unification of the Darwinian hypothesis. It's no longer a hypothesis, it's well studied. But this means in evolutionary practice that you can treat any disease or understand any problem, perhaps by studying some completely unrelated, apparently unrelated organism. And therefore, a standard view of many biologists that if you want to understand a particular, let's say, cancer, you must study that cancer and that cancer alone, is so obviously incorrect, because all the tools they use to study that cancer has come from studying other things. But we still don't seem to get that. So we must take an integrated approach. For example, Wnt is a molecule which causes many kinds of cancers. But Wnt was discovered at the Indian Agricultural Research Institute in an undergraduate genetic lab as wingless and in Drosophila, and proved to be enormously valuable later on because of the changes in molecular biology. Its value became known. So evolution links everything, and we must therefore be completely integrative in looking at all of biology and what we do. And that capacity has increased enormously with the access to big data. So our ability to extract information from evolutionary context everywhere is very valuable. So I would urge from an evolutionary perspective for companies like Immune to have a scientific advisory group to look at the mechanisms of which are involved in these kinds of immune, autoimmune processes in skin development and function, and so that the best information from all over the world is available. But let's come to development. Developmental biology is about how the parts are assembled to have function. Now, because of evolution, developmental biology, whether it's a worm or an elephant, or a bacteria or a plant or anything, uses a limited number of molecular mechanisms to execute the entire diversity of animals and plants that we see. In other words, the last stage elements of the toolbox are executed very differently in each organism, but the basic principles of short-term, long-term, and intermediate signaling apply everywhere. In other words, we can learn enormously by these signaling processes in multiple situations. And indeed, in skin, the kinds of signaling processes which are involved, we have learned in the nature of the immune system, we have learned from a variety of organisms because of a shared toolkit. So evolutionary and developmental biology come together. Now, the final aspect is function. And in function, I must also highlight one component which I've ignored in development. And development is an interaction between the lifestyle and the genetic material and the cellular environment. So to attribute everything to genes as a developmental process alone would be incorrect. But this is true of the lifestyle also. Development continues throughout life, and lifestyle is a major impact on how function takes place and what kind of resilience and what kind of homeostasis systems have. And therefore, when we look at apparent diseases which have components of both inheritance as well as lifestyle, the epidemiology of those diseases and studying that is very, very important. Now, from a practical point of view, I think it's correct and wise and sensible for companies to go for treatment, but they must have an antennae up to look at these three areas of evolution, development, and function, so that that treatment can be broadened or approaches can be broadened and used as technologies become available, as ideas also come along. So I don't suggest for a moment that companies, particularly smaller ones, should jump into every new technology or bandwagon which is there, but at least be aware of it and see how one can use that, whether it is RNAi or using inhibitors of various kinds or CRISPR and so on and so forth. Models to test it should be stimulated in other systems. Now, I'd like to end by, again, congratulating Immune about the daring you have taken up. But I'd like to point out one kind of a challenge. And you have overcome many of these kinds. Now, when you look at small molecules, because of the points I made about evolution, shared signaling pathways which are being used, and development and so on, whatever you choose will never be specific to the context you want. Because by definition, the same kinds of pathways which you're interfering with are used in multiple locations, multiple times during development. And therefore, the challenge is how can you impact on some context locally while the product you're using can have a global impact. Of course, the advantage with skin is topical, topical treatment is feasible. So that addresses a major component of that. But this is a very important problem in drug treatment and drug delivery, the specificity of location and timing. Second point in drug delivery is dose and context. Very often in biology, the kinds of molecules which are activators of pathways in one location are inhibitors of pathways in another location in another time. And this can happen in a dose-dependent manner, in a context-dependent manner. And therefore, again, there is a big challenge, not only in topical treatment, but in understanding that in the context of how various kinds of ups and downs in regulation can come. So here too, I would urge, while one looks at the efficacy and the safety of molecules which are being tried out, one should have a research division which looks at what it does in different contexts so that you can tweak the molecule and tweak the context as you need, not only now, but later. The last point, of course, is that these are called trials, and they're called trials because they can succeed or fail. And one should be very careful in not being overtly pessimistic and not try things out, or be overtly optimistic and assume that everything will work out. Trials have a low probability of success everywhere in the world, especially as you go further and further to advanced trials. You can cure almost every disease in an Eppendorf tube, or even many diseases in a mouse. But taking that to human context is a very big challenge. But it is something which one has to try out. So I would like to congratulate you. These are complex, presentations which are of greater importance to countries where there's a substantially pigmented population. And these are very, very important. Other companies, other countries are not likely to take it up. But there are still problems, even if others are taking it up. We should take it up in a courageous, innovative, and new way. We are a very large country with a large number of young people. And the glass half empty viewpoint is that every kind of problem in the world we will have on large scale. But the glass half full point of view is that we will be able to provide more than solutions, not only for us, but for all of the world for each of these problems. Thank you very much.
Thank you. Thank you so much, Professor Vijayaraghavan. I think we brought out so beautifully and interleaved many aspects for startups like us, and in a more generalized principles. That mechanism of drug action is a fundamental to the process that everybody should strive to figure out. Because as you rightly said, a molecule may have both positive and negative effects for different pathways, and that might eventually have an outcome issues and so on and so forth. But of course, the research is a key part. Many of the people at Ahamnune are trained in the research labs, of course, primarily, and that we will keep that in focus. And more importantly, you also made a very important aspect at the end, going forward, that these are trials, and definitely we have to exert caution in terms of how we interpret the data and how we would take this forward for next stages in that. So we are extremely grateful to you for your vision and very succinctly, very beautifully laying out what in some ways what Ahamnune should be doing down the road. Actually, I think very, very well said. Thank you so much. With that, I really request to Marvel to take over. Thank you.
Thank you.
Thank you.
Thank you. Thank you, Dr. Vijayaraghavan. It's really thought stimulating. It's the point to what you have put. Definitely, we will keep in mind. We'll try to keep those in mind as and when we go further, and try not to miss them. Thank you, Dr. Natarajan, for sharing the session. Now I'll proceed further with our next speaker, Dr. Archana Singh. Let me. Dr. Archana Singh is a principal scientist in the CSIR Institute of Genomic and Integrative Biology.
Mahesh, I'm sorry to interrupt. I'm
Yeah.
around. I'll listen to Archana's talk, but I'll shut my video off.
Okay, sir.
Thank you.
Thank you. Dr. Archana will be speaking on discovering a brighter side to the vitiligo, a researcher's perspective. She is a principal scientist and associate professor at IGIB Delhi, and she has published a number of research papers in reputed international peer-reviewed journals. The primary theme of Dr. Archana's laboratory is to understand the crosstalk between different lineages of the cells in skin tissue in vitiligo, a cosmetic disfiguring skin disease. In addition to directly evaluating patient-derived skin at the cellular and transcription level, her lab also utilizes primary cell cultures derived from patient skins and organotypic 3D skin culture to perform preclinical studies. Her lab recently demonstrated a strong anti-skin cancer signature in vitiligo and also delineated the molecular basis for a reduced incidence of skin cancer in subjects with vitiligo, despite the loss of UV shielding pigment melanin. It's very nice to hear something positive about the vitiligo. Over to you, Dr. Archana. We are really looking forward for a very exciting deep biology. Over to you.
Okay. Yeah, can you see my screen?
Yes, we can see your screen.
Okay. Thank you. Good afternoon, everybody. Vitiligo is often associated with psychological stress, emotional burden, and social stigma. But today, on World Vitiligo Day, I would like to bring to you a more positive, a much more brighter side to vitiligo. And I would like to, before I go to tell you what it is, I would like to thank the organizers at Ahamune for giving me this opportunity to present my work in this wonderful forum. So as I think Parul already mentioned that the loss of melanocyte leads to, so the loss of melanocytes in the skin leads to the visible depigmentation. And if we look at a high resolution microscopy, we see that in the normal skin, the skin is full of melanin, the black granules that you see here. Whereas in the vitiligo skin, we see that there is a complete loss of this melanin. And that leads to the visible depigmentation. So the function of melanin is not just to impart color to our skin. One of the most important aspects or the function that melanin provides is to shield the skin from the harmful effects of UV. And when I talk about the harmful effects of UV radiation, one of the most harmful effects is that it can actually cause skin cancer. So if you look at this, we see that as the pigmentation increases in an individual, the risk of developing skin cancer is significantly reduced. So higher the melanin pigment, it is actually better for the skin and it can protect the skin from incidence of skin cancer. Now, when we come to vitiligo, what we see is that we know that the pigmentation level is very, very lower compared to the ethnicity matched controls, people who have no vitiligo. And still, despite this loss of melanin, there are several research papers which suggest that there is a significantly reduced skin cancer risk in these patients compared to the healthy controls. Now, this is something which is very surprising. And we were also very interested to find out, what is the reason for this? Why individuals with vitiligo rarely develop skin cancers? And today I'll be sharing one of the main parts wherein we have discovered that a growth factor called SCF or stem cell factor is the one which could be actually doing this job of protecting the skin from incidence of skin cancer. So let me briefly take you to the function of stem cell factor. So keratinocytes secrete this growth factor, which is taken up by the melanocytes in the skin through its receptor called c-KIT. And the SCF, when it is taken up by the melanocyte, it actually leads to a lot of melanin biosynthesis. And that leads to more of this pigment synthesis and ultimately the transfer to the keratinocytes, and this protects the skin from UV radiations. However, as we already know that there is no melanocytes in the vitiligo skin, so there is no recipient for the SCF. And still, surprisingly, what we see is that the stem cell factor is increased at both mRNA as well as protein levels in the lesional skin, which is devoid of melanocyte. So this was very surprising. And we thought to take this further and find out if there is a reason for this. And the first experiment that we did was we took melanocytes and keratinocytes and treated them with recombinant SCF. So we treated it from exogenous sources, and we found that not only were the keratinocytes able to respond transcriptionally to the SCF, but they had a distinct response. They had a different set of genes regulated compared to what we saw in the melanocytes when both were treated with SCF. So that was very surprising, and it gave us an indication that yes, even the keratinocytes could respond to stem cell factor. So to finally, because our main question was whether SCF could be relevant in UVB mediated protection of skin. So we designed an experiment in which we took skin sample, cultured primary human epidermal keratinocytes, treated it with recombinant stem cell factor, and then exposed these cells to UVB radiation to mimic what a skin would go through, and then analyze the cells. So the typical analysis, what we did was, because UV is known to mediate DNA damage, mitochondrial dysfunction, as well as lipid metabolism changes, we studied all these three parameters and saw what SCF addition could change in these scenarios. So all the three scenarios I'll go through one by one. We have done multiple kinds of experiments, but in the interest of time, I'll be only talking about the ones which I have marked in blue. So coming to the DNA damage, so we have used this gamma H2AX as an indicator for DNA damage. So this is whenever the cells are exposed to some kind of DNA damaging agent, what happens is that in the cells, there is phosphorylation of one of the histones, and that can be detected using an antibody. So here, wherever there is, you see a green color, that means there is more DNA damage here. So when we treat cells with UV radiation, we see a lot of DNA damage, which is expected because UV is known to mediate DNA damage. And this was much more than the cells when they were not treated with any UV. But in the UV treatment itself, when SCF was added to these cells, the DNA damage was markedly reduced. As you can see here in the quantitation as well, that SCF was able to downregulate this DNA damage, was subsequently protect the cells from DNA damage. And not just that, even mitochondria are another organelle which are severely affected by UV radiations. So we saw that when UV was given to these cells without any SCF, there was a lot of fragmentation of the mitochondria, suggesting that they were not functional. And as soon as SCF was given to these cells in the presence of UV, the mitochondrial elongation was seen, which was similar to the no UV condition. That means the mitochondria were again getting rescued towards a healthier phenotype. And this was done using one of the dyes called MitoTracker Red, which stains the morphology, which indicates the morphology of the mitochondria. And the same was also seen with transmission electron microscopy. This is a high resolution microscopy technique, where again we saw that with UV, there were degenerated cristae in the mitochondria, which was significantly rescued as soon as SCF was added to these cells. So again, suggesting this, SCF was also rescuing the mitochondrial degeneration, which was mediated by UV. The third aspect, what is also very interesting in keratinocytes, is that UV radiation is known to increase accumulation of lipid droplets. So we have used a dye called BodyP, which labels all the lipid molecules in the cells. So this with UV exposure, cells are known to have these accumulation of lipid droplets, and it is detrimental to the cell's health. This is already known. But what we saw was, as soon as SCF was added to these UV irradiated cells, the lipid droplet accumulation was significantly, it came down significantly, and this is here in the quantitation as well. So SCF treatment was leading to a reduced number of lipid droplets. And not just that, concomitant with this data, we also found that a lipase called lipoprotein lipase was significantly increased upon addition of SCF and UVB compared to only UVB. So the lipid droplets were getting reduced in the presence of SCF, and that was actually happening because of the LPL upregulation. The lipase was getting upregulated. So the next thing was, so we have again stained the lipid droplets, and we have also quantitated the DNA damage in SCF treated cells, which are UV exposed. And as I have already shown you, the droplets, the lipid droplets are less and the DNA damage is also less. But if we inhibit the lipase, as I showed in the last slide, the lipase is upregulated in the SCF treated cells. But if you inhibit the lipase using a pharmacological inhibitor called orlistat, the protective effect of SCF is significantly reduced. So the DNA damage, which was less in presence of only SCF, is now more when you inhibit the lipase. So SCF mediated lipase upregulation is actually important for its protection from the UV mediated DNA damage. So I'll come to the final regulator that we have come to know in this pathway, and that is deacetylase called sirtuin-1. And what we found was that addition of SCF in the presence of UV led to a significant increase in the protein levels of sirtuin-1, which was significantly downregulated when the lipase inhibitor was added to the cells. So inhibition of lipase is detrimental to the sirtuin-1 levels also. And as another experiment, when we activated lipase using a pharmacological inhibitor, we saw that sirtuin-1 was upregulated. And to finally find that what is the final effector molecule in this pathway, which is leading to DNA protection from DNA damage, what we found was that similar to the SCF treatment here, which is leading to reduced DNA damage, even the lipase activation, if we independently activate lipase, that is also leading to lesser DNA damage. But as soon as we are inhibiting the sirtuin-1 using this inhibitor called EX527, it is a specific inhibitor of SIRT1. So even in the presence of activation of lipase, if we are inhibiting the SIRT1, then the protective effect of the lipase activity goes away. That means SIRT1 is the final effector molecule in this SCF-LPL SIRT1 pathway, and that is the one which is helping the cells cope up with the UV-mediated DNA damage, even during the absence of melanin in these cells. So to give a summary of what I have told today is that the SCF upregulation that we saw in the lesional skin, despite the absence of melanocyte, they were able to act on itself. So it was actually an autocrine signaling, and it led to upregulation of lipoprotein lipase at the mRNA level, and that led to SIRT1 upregulation. The final upregulation of sirtuin-1 led to lesser damage of mitochondria, lesser DNA damage, and downregulation of lipid droplets. And this lipid droplet was actually found to have a direct impact on the DNA damage as well. So SCF, which is usually known to act on melanocytes, we found a non-canonical role of this in vitiligo skin, that it could actually confer protection to keratinocyte when the melanocytes are not there. So it's kind of an adaptive response. And this story, the one that I have presented today, is under review right now, but we have recently published another signaling cascade, which suggests that there are some non-coding RNAs which are also involved in protecting the epidermis, the vitiligo epidermis. And again, very interestingly, the final molecule there also we have revealed is sirtuin-1. So sirtuin-1 seems to be the major downstream effector in protecting the amelanotic keratinocytes. And I would like to end this talk with an even positive note that it's not just the skin cancer which is diminished in vitiligo individuals, it is also the internal malignancy. The risk of internal malignancy is also severely less. It's very less in these patients. So that gives a really positive side to these individuals with vitiligo. At the end, I would like to thank my lab members, my students, Hemang, Anshu, Rohit, Manish, and Soumya, and most importantly, my clinical collaborators who provided the clinical material for conducting this study. Thank you so much for your attention.
Thank you, Dr. Archana. This is really a wonderful talk, very nice science behind it. And congratulations for publishing such nice paper. So I got, I'll take one or two questions before moving forward. So one, there was a general question, whether this anti-cancer signature found, it is only for the skin or it is translated to other tissues. I think partially you answered that this anti-malignancy.
So most of the studies, yes, they have initially, the epidemiological studies only looked at the skin cancer phenotype initially, but yes, the recent paper says that even the internal cancers are reduced, like lung cancer and kidney and colorectal cancer. The risk of these are severely diminished in these patients.
That's wonderful. And since you know that we have a lot of general papers, so one very good question it came is that LPL activator that you say reduces DNA damage, would you try to suggest them in like any sun protection cream or something like for general public?
Yes,
So what is your thought
that's
on
a very good question. We are actually working on it. So, so this NO1886, it's actually tried on mice models also, not for UV protection, for other things, and it is found to be safe. They have found it for lipid regulation and obesity related things, not for really skin protection. But yes, the safety data we have got from that study, and I think it can be easily taken up for general sunscreens, yes, of course.
Like SPF and all.
Yes.
Yes. Thank you once again, Dr. Archana. So we have many scientific questions also that maybe we'll take in some question answer session.
Thank you.
So thank you. We will move forward. Let me now invite Dr. Maya Tulpule. One second, please.
Hello.
Yeah. One second, please. 1 second. So Dr. Maya Tulpule is a practicing doctor, MS General Surgery. She is also a founding member for Sweta Association. She is DA and director of Sehwas Hospital, Pune. She is practicing since 1983. Today, she will be speaking on vitiligo, the patient's perspective. Dr. Maya Tulpule is panel consultant in Joshi Hospital, Sanjeevani Hospital, Tata Motors, LIC of India. As I said, she is a founder of Sweta Association, Vitiligo Self-Help Support Group, which was established in 2001. And as I said, it means she is starting this self-help support group before even we are starting the World Vitiligo Day, which was started in 2011. She is also the producer of award-winning film Neetal and the past president of IMA Pune. She is the president of Setu, a group of various health-related support group. And based on her extraordinary contribution to the field, she has been awarded by multiple groups like Santabai Kirloskar Award, Baya Karve Puraskar, Dr. Anita Smruti, Sangharsh Puraskar, Manav Dev Dharam Puraskar, Anandibai Joshi Puraskar, Vanchit Vikas and Jeevan Sangatna Puraskar. She also participated extensively in Vitiligo conference, like the first World Vitiligo Congress in Milan. She addressed the Keynote in Alliance du Vitiligo of France. She was the first World Dermatological Congress for Patient Organization, Vancouver, that also she attended, and the World Vitiligo Day Conference at Shimla. So now I invite Dr. Maya Tulpule. I'll share the screen. I will share her slide on her behalf. So just let me share her slide for a minute. Yes, Dr. Maya, you please proceed.
Good afternoon, and thank you very much for giving me this opportunity to share the audience with you. I am going to speak on patient's perspective. We have been listening to many technical things and developmental things, which really look encouraging to patients like us. Next, please. Next slide, please. Next slide, please.
Can't you see the next slide? I have actually moved it.
No, you jumped up. I want second slide.
Yeah,
Go back.
yeah,
Yes.
yes. Yeah, is it
There
visible?
are many questions in the mind of the patient. Yes, sir. There are many questions in the mind of patient. We call it autoimmune, but I feel the exact cause is still not unknown why the melanocytes are destroyed. As we don't know the cause, there is no prevention. And till the date, I feel there is no total cure, no 100% cure. So I hope autoimmune gives us some better medicines and there will be total cure. The treatment duration is very long. So patient keep on asking, like, see, if you take antibiotic, you get cured within 7 days or something. But how many days we are going to take this treatment? And they tend to discontinue the treatment. And because vitiligo is a cosmetic disorder as per WHO guidelines, it is not covered under insurance. So there is no reimbursement. Like you see, tacrolimus is 40-gram tacrolimus is 1,000 rupees. And melanocyte transplant also very costly. So people can't avail all these treatments. And also genetic transmission is not clear. So everybody is scared whether it will come in the next generation, whether we should marry this girl or do that. So these are all questions we have in our mind as patients. Next, please. So what are our common reactions? Denial. No, this is not a vitiligo. There is something else. Then there is a lot of fear, discomfort, concerns. These are, I think, common emotions all of us know. And people can go to the length of suicidal tendencies, basically because of the stigma attached to it. Depression is a common thing. There is a social segregation, and we know all these things. The reason behind is there is no 100% cure. I would like to quote the model of leprosy. When rifampicin was used in 1980 and leprosy could be cured to great extent and there were no disabilities, the stigma reduced a lot. So the key to all these things, to all the depression and other things is 100% cure. Next, please. So in our Southeast Asian countries, stigma is a huge problem. I think Thailand is the only country who doesn't have stigma because someone in the dynasty has vitiligo and they take it as a favor. But in the rest of the countries, a huge stigma. When I speak about stigma, it is social stigma. That is what we usually talk about. But I will also talk about the personal or individual stigma. As a person who has gone through all this, I will say if social stigma is 40%, the stigma in the mind and eyes and behavior of the person who is having vitiligo is 60%. There is always negative self-assessment and a lot of problems leading because of it. Next, please. So how to cope with all these things? Everyone knows that self-acceptance is the best treatment, but it is not at all easy. Why me to, why, it's okay, is a very long journey, very difficult journey. But yes, you have to cope up with it. And not only the self acceptance, family acceptance is also important, because family support is very much needed. So how you can get the support from family? As I said, friends also can help you, colleagues where you work or you study, they are also important. And also the empathetic dermatologist, the person who treats you also should be very empathetic. Yes, next please. So here comes the role of self-help support groups. Sharing and caring leads to a lot of catharsis of emotions, and it makes the patient comfortable. Then we try to make their day-to-day life comfortable. Many people don't know that sunscreen should be used or cosmetic camouflage can be used. So we can make them comfortable like that. Then family issues are also there. Many times monetary issues are there, or disputes are there amongst siblings also. And marriage is a great issue, at least in our country. So we are helping for marriages also by developing a marriage bureau for last 20 years. So also sometimes there is discrimination in education or employment. Front desk jobs are not at all offered to these people, obviously, where looks are important. So we also help in those aspects. And we hope that no one should die of without food. So similarly, no one should be denied best possible treatment as far as vitiligo is concerned. And we also do a lot of social awareness to remove stigma and try to reach the people. Advocacy is our main object. So how we do it? Basically counseling by the person who has vitiligo, we found is more important. If a dermatologist goes and counsels, people say, "Aapko kya samajhta?" What we are going through it. So the person who is having vitiligo, if he counsels, it has great impact. Then we have conducted almost 55 matrimonial meets and more than 1,500 people got married. We have employment bureau also. We provide cosmetic camouflage. And recently we have set up these phototherapy units, narrowband UVB and excimer laser at very affordable cost. And we also publish a lot of pamphlets on treatments, what to avoid, then phototherapy, then regarding diet, because there are many misconceptions. So we do all those things. And side by side, we are doing social awareness programs also. We conduct 2 meetings at least per year. Then we are conducting Run for Vitiligo marathon for last 7 years. And cycle rallies, publishing newspapers, essay competitions, etc. Thanks. Next. So we have been associated with National Institute of Immunology, with Dr. Rajesh Gokhale and Dr. Rajni Rani for a long time. And I wish that something good comes out of it. We always are away, we are very eager to volunteer for any trial. Parul, you can listen to it and encourage in research. And such webinars are a ray of hope for us. Again, VPOC also, where Vitiligo International Patient Organization Committee, in which I am also working. So they are also going to listen to this video because everybody is eager to know what new we are having. So I again appeal to all the listeners that we need donations. We have no government funding. If we go to health department, they say this is not contagious and this doesn't produce any disability, physical disability. So why you need to give some assistance? But my appeal to researchers is if you can find a vaccination for COVID-19 in a year, you can do something great for us also. So you can at least inform the people who are having vitiligo that there is such self-help group working for 21 years, and we need some volunteers also. So this is my earnest appeal to you. And our movie Nital will be on YouTube from today onwards. So anybody interested can watch us. Next, sir, please. And this is how you contact us. Thank you very much. So nobody is perfect in the world. So you are perfect exactly as you are. So you should love your body, and there should be no body shaming for whatever flaws you have. That is what I feel. Thank you very much.
Thank you, Dr. Maya. Thanks for providing the social or psychological side for the vitiligo. We like working in the science. Sometimes we get too much engrossed in the science or the pathways and all that link to that social stigma we sometimes forget. So actually I got a lot of questions for you. Maybe we'll have that in the question and answer session because we are rightly on the time. So we have a next speaker is Dr. Devendra Prasad, and after that we have a question and answer session. So in that we will ask, because our audience, many of them are the general public, so they have a lot of general questions. So let me first welcome Dr. Devendra Prasad. Let me introduce him first. Though people who are working in the field, they know him very well. Dr. Prasad, let me share the screen for a moment for your introduction.
Okay.
Yeah. Yeah. So our last speaker for today's session, Dr. Devendra Prasad. He is currently working as a professor at PGI MER in Chandigarh. He's he's also a professor of dermatology, University of Rome, Jim Marconi. He has around 30 years of experience in the field, and he has published more than 290 papers in various journals, out of which 140 are on vitiligo only. He has written several chapters, major textbook. He has attended more than 150 international conferences and meetings. And because of his tremendous contribution in the field, he has received many awards and adoration for his work, such as Best Clinician Award by Himalayan Institute of Medical Sciences, and MBD oration of Indian Association of Dermatologists, Venereologists, and Leprologists. He was also the president of the Asian Society for Pigment Cell Research from 2011 to 14. He is also a council member of International Federation of Pigment Cell Societies. He is an associate editor for Dermatology Therapy, Archive of Dermatology Research. He is a member of many professional bodies such as American Academy of Dermatology, European Academy of Dermatology and Mineralogy, Asian Society for Pigment Cell Research, he is president for that, Society for Investigative Dermatology, Indian Association of Dermatologists, Venereologists and Leprologists, council member of International Federation of Pigment Cell Society. He's working on many research projects as well, other than being a clinician. Like few of them are the functional characterization of Al-Aqsa alpha transcription.
Prasad, thank you for taking time out to come here.
Yeah.
I mean, everybody knows you in the vitiligo field. I think no introduction will be enough for you, but please start here. We're very eagerly waiting for your talk. Please start.
Thank you. Thank you, Dr. Parul. Thank you, sir, for kind introduction. So I'm happy to be here and thanks for organizing this webinar on World Vitiligo Day. So this is being organized all over the world to sort of emphasize the importance of vitiligo. So this patch sums up story of vitiligo. So like for dermatologists, this is an acquired depigmentation disorder devoid of melanocytes, but from patients' perspective, it is their worst patch. How smaller it may be, but small patch can have a huge stigma, and it can lead to the impairment of quality of life, as Dr. Maya has already nicely presented this part. So what probably in pathomechanism, so many things are happening, starting from autoimmunity, still the mainstay, but other things like altered cytokine milieux, defective keratinocytes, fibroblasts, growth factors, so ultimately lead to the absent melanocyte in this patch. So this might have been already been covered. So sort of, it can be said that this is a dance between three partners. So there's a genetic risk is there, and in the presence of triggering factors, the autoimmunity sets in that leads to the damage of melanocytes, finally resulting in this depigmentation disorder, which we know as vitiligo. So this is probably the convergence theory, and finally, this may be some melanocytorrhea, certain melanocyte defects, oxidative stress, and finally this pathway of CXCL9, 10 interferon gamma, and TNF-alpha pathways emerging as one of the major pathways, so which has a lot of clinical translation. And we know the JAK inhibitors, they are acting through this pathway. So they are going to be, I think, a very important modality in the management of vitiligo in near future. Just from clinicians' point, so there is sort of a fight occurring between the good T cells versus bad T cells. So the balance is tilted toward the T effector cells. So as of now, all our modalities are focusing at removing these cytotoxic T cells with immunosuppressive agent. So the future treatment option would be like, can we attract the good T cells to the patch so that the balance is tilted toward the good T cells without using immunosuppressive? So probably like CCL22 activation can attract this T regulatory cell to the patch. So these are the future options in the management of vitiligo. And moreover, this stability is very dynamic process, especially in non-segmental vitiligo. So even if the patches are successfully repigmented, they can show the depigmentation at the same site, maybe after 15 to 20 years. This is because of persistence of cytotoxic memory CD8 cells or T8 cells. So why they are persisting? So this could be through this pathway of interleukin 15 and CD120 pathway. So John Harris and other Julian, they are working on this pathway. So if we block this interleukin 15 or its receptor CD122, so it can lead to sort of a lifelong stability in vitiligo. So this is very interesting. This is paper published this year from the Chinese group in the Nature. So this was, we were wondering all the time, like acrofacial vitiligo used to be slowly spreading. So it's very difficult to stabilize acrofacial vitiligo. And moreover, the response to treatment was not very good on this acrofacial areas. So there were many hypotheses working on this, why this so that acrofacial lesions are so resistant and they're so even resistant to stabilization. In this paper, the author have shown that there are distinct fibroblast subset, and probably the fibroblast in the acrofacial area, they can augment the response to this interferon gamma, CXCL9, CXCL10 pathways. So this is very interesting. So in future, we need to target those fibroblasts. We were now focusing just on melanocytes in the vitiligo, but this is not the case. So all the cells are defective. So we need to target those cells as well. So this is just to make this repigmentation fast pathway so we can act through the Wnt pathway. So this is a study by our friend Thierry Pesseron from France, where they have shown that if we can augment this pathway by using agonists, so we can help this repigmentation faster. So I'm just talking about the future treatment. So in our lab also, we have working on one of this agonist of Wnt pathway. And as you can see in this mouse model, the topical application of this molecule will lead to the better and quick repigmentation. So just to coming to the clinical part. So when patient present to us, so we need to know what is our aim of treatment. Is it stabilization? Is it repigmentation? If patient present to us with evolving new lesions, rapidly spreading lesions, then first we need to stabilize the disease with certain group of drugs. And if patient of stable vitiligo present to us, then we have to focus on repigmentation strategies. And finally, when patient doesn't respond to the medical management, we move on to the surgical management. Ultimately, our aim is the complete repigmentation and total stability, which is that an ideal treatment of vitiligo. And I think our journey toward this ultimate aim is, I think, very going in a right direction as of now. So what we can offer to our patient to stabilize the treatment. So first, I just want to highlight that all the vitiligo should not be treated like a same, like the patient comes and we give it just a one standard treatment to all vitiligo patients. This is not the case. We have to first analyze if patient has present to us at early stage, what is our objective? Is it stabilization or is it repigmentation? And then we formulate our strategy accordingly. So as I mentioned, there's a huge data on oxidative stress in vitiligo. So antioxidants should be effective. And as such, there are no many clinical trials on the role of antioxidant. Good evidence-based studies are lacking, but still based, because we know in vitro, like there's a huge oxidative stress. So why not use antioxidant? Because they are quite safe. So we in our clinic, we use it in an early stage or as a maintenance therapy. I mentioned watchman therapy, as you say in a cricket match, like if just we send that, some lower order batsman towards the fag end of the day to see if they can survive the attack of bowler. So similarly, we can use this at an early stage to see if they, because they are safe, but if they can control the slow spreading activity. And we have used oral ginkgo biloba as an antioxidant. And there are other antioxidants like polypodium lichiotomas, some beta carotene and combination of certain vitamins. So they're quite safe. And superoxide dismutase has also been used because it is also a good antioxidant. And we have used minocycline as, again, as an antioxidant because there was one study where they have shown that minocycline protects the melanocyte against the oxidative damage. So minocycline in a dose of 100 milligram per day can be used in slowly spreading vitiligo. So there is more and more paper are now coming on antioxidant. Again, one paper from Thierry Pesseron from France, they have shown that gliadin protected superoxide dismutase is very effective and combined with phototherapy. So we hope like in near future, we have much better data to support the use of antioxidant in the management of vitiligo. So as of now, this oral mini pulse therapy remains the mainstay of controlling the disease activity. Here we give low doses of steroid, like 2.5 milligram of dexamethasone on 2 consecutive days per week. So we have seen, like we have now experience of using this in more than 20,000 patients, and we can stabilize the disease in more than 80%. But the question remains like how long, because it's difficult to predict how long we need to give it. So what we do in our clinic, we give it a sort of hit and trial. So because after every 3 months, we try to taper it or stop it and see if the activity is controlled. If not, then we give another course of 3 months. This is low doses. They are quite safe, but still they have side effects. So patient need to be monitored. Similarly, we have shown this a low dose of methotrexate. So it's again shown to be effective in the management of controlling the disease activity. And similarly, one can use azathioprine, mycophenolate mofetil, cyclosporin. So they're all broad spectrum immunosuppressive agents. So they can be used to control the disease activity if patient present to us with rapidly spreading vitiligo. So now, as I mentioned, this pathway of CXCL10, CXCL9, so if interferon gamma, so JAK inhibitors act through this pathway and control this pathway. So this as good as no vitiligo. So there's quite data is coming up. And initially there were a few case reports of tofacitinib, ruxolitinib. Now we know this ruxolitinib topical has been tried in a phase 3 trial. So this was published in the Lancet. So this is again from our friend Hamjabi's group. So they have shown that 1.5% ruxolitinib cream is quite effective, and they have shown in further stratification of data, like even long-standing vitiligo, they have responded to topical ruxolitinib cream. So this is, I think we are watching this molecule very closely. So it's going to replace our existing modality, topical modality at least. So they have nicely again correlated with this pathomechanism. So this is for this clinical translation. So is ruxolitinib is blocking this JAK inhibitor pathways through CXCL9, CXCL10, and 1.5% twice daily application resulted in very significant improvement of FBASI even in patients who have not responded to the phototherapy. So there are few another analogs there in phase 2 or phase 3 trials like tofacitinib, ritlecitinib, baricitinib. So I think that future for vitiligo is bright. We are getting some vitiligo specific molecule. So just to touch upon our second aim of repigmentation, once we have stabilized the vitiligo, so then we start focusing on repigmentation because it's entirely different process. So there are lack of melanocyte. So we have taken care of the mechanism which were damaging the melanocyte. Then the second aim is how to make them proliferate faster so that repigmentation become faster. So phototherapy remains the mainstay. I have not touched in this presentation narrowband UVB therapy, which is the mainstay, which act through both the pathways, stabilizes the disease as well as repigment. So this remains the gold standard as of now. So what else we can offer along with this topical steroid, topical TCI inhibitor, certain growth factor, prostaglandin analog that can be used alone for a smaller patches, and if extensive can be combined with narrowband UVB therapy. So this is just to show like this is sort of lot of crosstalk going on among these cells, keratinocytes, fibroblasts. So this is just not the melanocytes that are defective. So various keratinocytes and fibroblasts are defective. They are not able to secrete the good growth factor for melanocytes. So that can result in more damage to the melanocyte. So make the melanocyte more prone to the autoimmune damage. So this is, we know this alpha MSH-MLR has been tried as a melanocyte 16 milligram subcutaneous monthly implant. So this is data by Pearl Grimes group. They have shown that when combined with phototherapy, this 16 milligram injection monthly can increase the speed of repigmentation and finally the better outcome of repigmentation. Similarly, in India, we have some experience of using this topical BFGF, which can again combined with phototherapy, or it can be combined with topical tacrolimus. So it, as you see here, it can also give repigmentation of certain resistant patches with leukotrichia also, because it can induce marginal type of repigmentation by attracting the melanocyte from the border of the patch. So we have shown in this paper, like when we combine this BFGF with tacrolimus, it worked better than tacrolimus alone. So another group of drug is prostaglandin analog. They are again for repigmentation. So this I just want to highlight again that we should first look what is our aim. So if there is a stable patch of vitiligo not showing good repigmentation, we can try these type of modalities. So initially we have done this study on this prostaglandin E2, and then now we have more data on other preparation like bimetopros and latanoprost. Like in this patient, there was loss of eyelashes, that is alopecia areata was there, plus depigmentation, and patient on users of this latanoprost eye drops, there was regrowth of eyelashes as well as repigmentation. So what is the issue here is like we are using ophthalmological preparation for skin also, but that is not an optimum preparation. So I understand some of these companies are coming up with the dermatological formulations of this prostaglandin analog, which are again good molecule for the treatment of vitiligo. So what's new on the vitiligo treatment horizon is JAK inhibitors, oral and topical. So we are eagerly awaiting more data on this, and I think cost is limiting factor as of now, but I think in near future is going to be one of the important modality, the JAK inhibitors. So interleukin-15 blockers, again, for inducing a sort of permanent stability, and CD122 blocker acting through the same, and through the CXCR3B interaction, which whereby even we can act early before this autoimmunity sets in if we act at this level of CXCR3B interactions. Thank you.
Thank you, Dr. Prasad. It's really very nice presentation. You touched all part of the vitiligo. And first of all, let me thank you being a doctor and doing the research. This is a combination we generally don't see too much in India. So you can see from the patient side also, you can see the actual what is happening, and as well as you know about the research, what is going on in the deep. So thank you for doing that.
Thank you.
So we'll start our question and answer session. So first I'll start with you only, Dr. Prasad.
Okay.
You talked about when we talk to patient and when they talk to their doctor, so people have two type of thought. One is that they don't have any medicine for the vitiligo. This is the come. If I got vitiligo, I don't have any medicine. Second is that, like you described, there are so many things, there are so many hopes of the patient. If it can be carefully formulated, one can achieve good result. But the patients are confused because if they go to take a second opinion from the doctor and the treatment is entirely different. So do you want that any strategies there which can be taken as a country basis, or how should we go ahead for that?
Yes, that's very interesting question. So this is what we are working on. I mean, now the situation is much better. When we started way back in 1993, so there was question to treat or not to treat, because even a Western, like especially dermatologists from other Western countries, so they were like saying it's a cosmetic disease, so need not to be treated. So, but this is now maybe the gradual over the years. So we have like global vitiligo working group, European task force on vitiligo. So this has become very important disease for maybe for last few years, and even from countries like US or Europe. So they are now focusing on vitiligo. And that's why like many pharma companies, because earlier we used to borrow these molecules from based on our knowledge from treating from other autoimmune diseases like alopecia areata or psoriasis. So that can be used for vitiligo. But now I think this is time has come now where pharmas, has so much interested. So vitiligo specific molecules are getting attention. So I always counsel my patient, like this is sort of a phase where we expect a near breakthrough in near future. So one should be very optimistic. And you rightly mentioned, so some of the dermatologists, they still think like this is nothing much can be offered. But that's why this World Vitiligo Day, how it started about, I think in 2011 or 12 maybe. So and then start focusing on this. So with the two aim, one is patient education, and second is among all dermatologists or treating physicians, pharmaceutical to sensitize all these group to, just get some good molecule, because this disease has a quality of life impairment, which may be, I think, worse among all skin diseases, I would say.
Okay, and I have one very basic scientific question, like the CXCL9, 10, and other cytokines which are higher. Are those seen any Indian scenario, Indian patient, they are also higher? Any data like that?
Yes, we have data. So they have emerged a good biomarker. So there are many studies coming up from India where they have shown that this can be a good biomarkers for the activity. It has been shown that CXCL9, CXCL10, and it's correlated with the disease activity.
Okay. And so one last question to you. Like Dr. Kiran Marthak pointed that if a therapy is very like safe, it is still advisable even if the efficacy is low. So it may take me a little more time, but if it is safe. So nowadays, like new therapies like JAK inhibitor, capacity inhibitor, ruxolitinib are coming. Some of them have a black box warning already there. And we know they were not developed for the vitiligo, they were developed for something else. Now they are positioning it. So how do you think that those type of medicine, which is a tremendous potential for side effect and to go ahead in the country like India, where some person can prescribe it a very high dose for a long time. So what are your thoughts on this?
Yes, so they are very good drug. Again, like you said, there are some concerns. And if proper protocol followed, so one can properly monitor those patients. So this would be very safe and effective, but that should be, done. It should not be like if everyone start prescribing it without monitoring the patient. So they are concerned regarding that. But now FDA approved study would be there. So for specifically for vitiligo, so that would be again, which could be, pinpoint about the safety and efficacy in vitiligo also, and one can safely prescribe. So as for our experience, we have started using this in our patient with proper monitoring and found it to be better than the other immunosuppressive, broad spectrum immunosuppressive agent, safe device.
Thank you very much. Thanks a lot, sir. So one question I'm asking to all the panelists here, we are keep getting is that food habit, how it changes. Like many people say that the eating fish and milk together can lead to it. So generally it's taken as a myth, but I think if some of you speak to the audience and tell about not to worry about it, it's good. So sir, either you or Dr. Maya Tulpule or anybody in the panel can comment on it. It'd be very nice.
Yes, with patients, people feel that white things like milk, rice, banana, and curd is also bad. Then virudhar, like you were talking about with fish and curds or fruits and milk and fruit salads, they are all bad. But I feel there is no scientific basis for this. And many people come to us following all this and they get so cachexic, exhausted, their hemoglobin is low, their nutrition is low because they have stopped all this. And another thing I would like to point out is in India, percentage of vitiligo is supposed to be more in Gujarati people.
Yes.
And they, for thousands of years, they have not eaten fish or anything.
Yes.
So this is all a myth and nobody should fall for it.
Yes. Okay, and as I said, Dr. Maya, I have a lot of questions from the social side of it. Like many people, they say, like, I have a vitiligo, what will happen to my sister? Right. So it's difficult to answer based on the scientific data. So what message you want to give to them?
No, there is no definite genetic pattern for vitiligo, as we do not know the gene, so we don't know the transmission. The gene is supposed to be partially penetrating or dominant, but nobody can predict that. A lot of studies are going on, but still not a single gene is found. So Ayurveda says that the third generation can get it. But even that is not true because we have data of about 1,000 people. No, it is not true. So if vitiligo percentage is 1 or 2, I feel 80-85% can people get it anytime at any age and when there is no history of vitiligo in the family. So this misconception should be removed first, and it is a big hindrance as far as marriage is concerned. Because people feel that we don't want to introduce such gene. हमारे घराने में ऐसा कुछ नहीं है. We don't want to marry such girls or a girl or boy from this family. So, and it is that a lot of social education is needed on this aspect.
Yes, that is a very important question. So many of our patient comes to us like before, if someone found that someone in family has vitiligo, so there's a lot of meetings going on. So this is very tricky question. If you say a clear cut that there will be no risk of vitiligo, it's difficult to say because there is genetic risk is there. So the proper answer to this, we usually based on scientific evidence that Richard Spears, I mean, we always used to question him about this because this is the most important question we get in here in India. Like, what would be the chances if one in the family, like one mother or father has vitiligo? So what would be the chances of developing vitiligo in the kids? So they are very concerned about that. So there's a clear data like in monozygotic twins. So there's chances are 23%. So just 23% in monozygotic twins, and otherwise the chances in Caucasian skin is around 6%, and according to him in like in Indian skin it would be like around 4.5 to 5% genetic risk. So as Dr. Maya mentioned, this prevalence is 1 to 2%. So we explain to the patient like I have a risk like 1 to 2% risk of developing vitiligo, and otherwise if one parent has, so it is just like 5 times, so just 2 to 3 times more, so it's very safe otherwise. So 95% chances are there will be no vitiligo, but we can't predict who in that falls in that 5% bracket. So there's just slightly enhanced risk of developing vitiligo, but this is not as such. But the myth is like one, so they don't marry even if one distant relative, blood relative has vitiligo. So definitely this genetic risk is there, but it's of very limited value.
Okay. Thanks. Thank you, sir. And thank you, Dr. Maya. So my next question is to Dr. Martha, sir. You have done a lot of clinical and trial for many type of different therapies. Now you have seen like Dr. Prasad is speaking about the new modality or the treatment which is already there and the new treatment which are coming. And each treatment has its own sort of like say, side effect and all. So just purely from the scientific basis, for the patient benefit, this and keeping that in for the psychological situation of the patient in mind, what will you suggest that how to make a proper balance for that? Any thought on it, Dr. Martha? I can't hear you, sir. Can you please unmute you?
Can you hear me?
Yes.
Yeah. As Dr. Prasad also showed that there are multiple treatments are available, but there cannot be a standard treatment which is true for all the patients. Some can tolerate something, another will tolerate something else. So it cannot be one cap can fit all the heads. So it will vary from patient to patient depending upon the age, the sex, the other ailments that they are suffering from, genetical history, hereditary conditions, etc. So it's very difficult to opine that a particular drug is the best. But of course, the at present, which is more or less like a standard of care, is the phototherapy. I think is one of the best treatment that is being offered. But at the same time, one has to be careful, even with phototherapy. So it's something which is good for me may not be good for somebody else. So it depends on every individual who takes the treatment. So certain time, if there is a particular product which is causing any adverse effect or not giving a desired response, one may need to shift over to the other line of treatment. So it will vary from patient to patient, in short.
Great. Thanks. Thank you so much for answering that. So we have a lot of other questions. So just for the sake of the time, I'll just ask one question to Dr. Archana. So Dr. Archana, even though I myself has a lot of scientific questions, which I'll take later. So just one question pop up is that vitiligo is caused by autoimmune destruction, right? There is also some genetic predisposition.
Yes.
So what did the protection from cancer you're talking about? Is it because of the high immune background or any genetic factor responsible for it? Or is it some other factor? What you think
So
about?
the studies which have come up which say that there is a reduced incidence of internal malignancies in these patients. They actually say that these are because of the autoimmune responses actually.
Okay.
The immune milieu is so perturbed that it is actually now having a good impact on protection from cancers. So the autoimmune component is actually playing a role there. Nobody knows the actual phenomena, but they suspect that the immune response has is gone haywire, but in a positive way for cancer.
Good. I'll catch up you with again. So since we already exceeded the time, right now I'll request Dr. Parul for the vote of thanks for the conclude the session. Over to you.
Thank you, Mahesh. I think this session can go on for another couple of hours. We're having such interesting discussions, and I also want to but maybe that's for some other time. But needless to say, we had an extremely empowering and an enriching session. Thank you, all the panelists. I think I learned every time I talk to all of you, I learned so many new things. So thank you all so much for coming and taking out time. I think there's so many take-home messages. If I have to just encompass it in a couple of sentences, I think what we as a company have learned is that we need to focus a lot, as Dr. Vijayaraghavan said, we need to focus on biology in an integrated manner, not look at it in a separated, isolated manner. And Dr. Prasad, thank you for giving us so many new information about research, clinical research that is happening about vitiligo. People must be really excited. As I always say, this is an exciting time for vitiligo, and people should be looking forward to a solution to this problem very soon. Dr. Archana, again, the brightest